So this study is using H&E slide, and that slide alone. We're not doing any special staining. This is just a simple everyday H&E slide we generate for any tumor diagnosis. So we used that H&E slide, whole slide imaging, and used AI analysis to classify these tumors into 3 distinct phenotypes, immune phenotypes, and those are, as you well know, inflamed, excluded, and desert, hot and cold tumors. So we made that classification by doing manual training, like we identified lymphocytes inside H&E slide and trained them do the machine learning to have AI figure out what are the percentage, the immune score, or the lymphocyte, we call it TIL, right? So TIL score of these H&E slides, and also used the distribution of the stroma and tumor and the location of lymphocytes to figure out the 3 distinct immune phenotypes. So we did that analysis in 383 samples of differentiated thyroid cancers coming from the TCGA data set. And what was interesting is what was typically thought of as a very cold tumor did have a lot of inflamed signatures here. The deserts were 47%, excluded were 34%, and 17% were inflamed phenotype. So these are potential opportunities for immunotherapy response even in this differentiated thyroid cancer. So we thought this was the most meaningful finding, and we sort of looked at its relationship with many other clinical or pathologic factors or survival. But what was noted was we thought that BRAF mutation, especially V600E mutation, or the tumor differentiation score, although they're all well differentiated, but their differentiation level is a little different. So we look to see whether those have any relationship with these 3 distinct immune phenotypes, and the answer is no. They did not have any significant correlation with these factors, but rather proving that these immune classifier can be a distinct, independent predictor of response for possible immunotherapy. And there are clinical data out there that these groups can still be target. This histology can be still areas of immunotherapy response. And in DART study, we do have, with the SWOG 1609 study, we do have thyroid cohort. We did present this in a national meeting and showed nice, interesting responses with Devo-Ipi in these thyroid cancers. So the take-home message is really that more than 15% of differentiated thyroid cancers can have inflamed phenotype, and this is a new opportunity for immunotherapy in this high unmet need cancer.